Coastal wetlands of the St. Lawrence River are facing a lot of challenges, and these have been growing over the years. So, of course, climate change encompasses a lot of things, but there's issues such as water levels, water quality, there's contaminants, siltation, different water clarity is another issue as well. We have invasive species moving into the St. Lawrence River and moving into coastal wetlands. This includes vegetation, invasive plant species, as well as invasive fish species and vertebrates. We have a lot of habitat loss happening with wetlands. They're, of course, tend to be along the shoreline. This is really highly prized real estate, so a lot of people want to develop that, and it's resulting in the loss of wetlands in a lot of different areas. Wetlands, the last point there, food webs. Wetlands are one of the most, if the most biodiverse types of habitats you can find in North America. So they're extremely critical for preserving, just to preserve biodiversity. If you had to pick any type of habitat, you would most likely pick wetlands or a few others versus, say, a field or even forests, for example. Wetlands are home to so many different species of plants, animals, and vertebrates, and different life stages of these animals as well. They could be living in wetlands for part of their life cycle, for example, if they're just born or if they're spawned there. They're really critical types of wetlands. So it's really important that we study wetlands, we preserve them, and we fill in knowledge gaps as well. There's a lot of things we don't know about wetlands. We don't know specifically what species sometimes live in them, or if some have disappeared from wetlands, how they're using them. So there's lots that we need to know about wetlands in order to preserve and make sure that they're remaining healthy. So the FINS project is one of these really large-scale projects that we have on the go at the River Institute. And it was started in 2015, and it's a partnership between the Mohawk Council of Akwesasne and the River Institute. So back in 2015, both organizations got together and realized there wasn't a lot of information on small fish populations of the river, many of which inhabit wetlands as part of their life cycle. So to fill in this knowledge gap, we started this project where we go out in the river. You can see two team members here doing some seining to collect fish in these nearshore environments. There's a nice wetland there. They're in that picture there. And we wanted to collect information on what fish are in the river and all across a range of different habitats. We also wanted to collect information on the actual characteristics of those habitats. So what plant species are there, what types of sediments are at the bottom of the river, what the water quality was like in different types of habitats, and I've listed a few there. This is also a great project where we're able to document species at risk up and down the river, hundreds of kilometers of stretch of the St. Lawrence River where we're studying. So we're really adding some valuable information on the distribution of some key species that actually make wetlands their habitat as well, specifically fish species. And we're also documenting the occurrence and distribution of invasive species. So this could be plants, aquatic plants underwater, terrestrial plants above water, and also fish. So a nice picture. There's some different team members there working together. And again, we've been working together since 2015. This is a really great partnership between our two organizations, and we're keeping it going. So we're going to be out again in 2021. Continuing it. This map here just shows the overall extent of the project. So it's huge. This is almost 300-kilometer stretch of the upper St. Lawrence River. So where the Lake Ontario transitions in the St. Lawrence River all the way down to Montreal. And the project has also now extended up from the St. Lawrence River into the Ottawa River. So we're expanding downstream and also up into the Ottawa River. The different things, I'll just go through some slides showing the types of information we're collecting as part of this project. And this also relates to information on wetlands. So we're in terms of fish communities, we're looking at how many fish are at a certain site, species richness, so the diversity of different types of species. And it really does range across different habitats. In some cases, we might only find 2 or 3 species at a site. In other ones, we might find over 20 species. And the sites where we find over 20 species, we really want to study closely and figure out why it's supporting such a rich biodiverse fish community. We also take morphometrics, just a word that means we're measuring different characteristics, physical characteristics of the fish. most notably fish lengths. Here's a nice picture. So we have a smallmouth and largemouth young of the year bass here. Largemouth bass, young of the year, very common species found in wetlands of the St. Lawrence River. In some cases, super abundant. We're also measuring aquatic habitat characteristics. So the bottom of the river, the substrate types. In some cases, it's very silty in some sites. In other cases, it's rocky. And that really builds your habitat from the ground up and determines what kind of plants can live there, and then also what kind of fish are going to be happy living there. So water quality as well, and aquatic vegetation. So in some cases, in some habitats, you can see the surface of the water is covered in certain types of vegetation, like duckweed. Water quality is something we measure at all of our sites too, so just basic things that really help us understand why certain fish assemblages and species are living at those sites. So temperature, fish are cold-blooded species, so they're really strongly influenced by temperature. They can't tolerate certain temperature ranges above or below. So that can determine the distribution of fish. Water clarity is also really important. Some fish really need crystal clear water, and they're very intolerant of muddy water or cloudy water, which can be caused by siltation, runoff, activities in the water too that stir up the sediments into the water column. We also take measurements on nutrients. So phosphorus is one and nitrates that run off from the landscape, and you see a lot of that running into tributaries that are flowing by agricultural fields, for example, that flow into the St. Lawrence River. And when you have lots of nutrients, you could have algal blooms, you could have a lot of plant growth in some areas, and then that could affect the fish in that area as well. So all these are different, really important factors for measuring to help us understand where these small nearshore fish populations are and why they're there. We're also looking at the actual shoreline characteristics, and if anyone watching has ever been in a boat and traveling up and down the St. Lawrence River, we're quite fortunate in the stretch around Cornwall, where you have lots of natural shoreline still, particularly in the Akwesasne territory. There's some beautiful shorelines, and if you go upstream of Cornwall towards Brockville, there's quite a bit of developed shoreline, and again, downstream of Cornwall into Lake St. Francis, particularly in Quebec, most of the shoreline has been developed except for a few patches here and there near some wetlands. So it's something that we measure, and we try to figure out if the development of the shoreline is having an impact on fish communities and the health of the aquatic ecosystem immediately next to those shorelines, if they've been developed or not. So it's something we record. We record what types of plant communities are on shorelines as well. That's a nice picture. I'll go back there. It's a nice picture of a grass pickerel. So that's a species of risk that inhabits particularly wetlands. And in this case, this is a wetland near Akwesasne. Our project is providing extremely valuable information on the distribution of species of risk that didn't exist before. So I've just listed a few here. There's some fish species of risk. We're also noting when we come across freshwater mussel species at our sites, also plant species or vertebrates like turtles or amphibians that we see. We record all that as well. And all this information gets reported to provincial and federal sources, and then goes into those databases and then helps expand the known distribution maps of these species. So it helps us keep track of whether these species are doing well, if we're providing new sites where they are, or if we're visiting sites where they've been known to occur previously and we don't find them anymore, then that could be a big problem. So what will be collected since 2015? Quite a bit of information, actually. So over the last 6 years, 6 field seasons, we've collected we've documented over 130,000 fish in the St. Lawrence River. So this is a huge data set now. Over 200 sites we visited. You can see all those blue dots are sites where our crews have visited. So that's all the way down Wolf Island by Kingston and then flowing downstream past Brockville. Lots of sites in the Cornwall area, so immediately upstream and downstream of Cornwall, we have that really well documented. And you can see our sites now are expanding downstream towards Montreal and then a little bit up into the Ottawa River as well. So those are kind of frontiers of where we're trying to move And the more geographic expansion and coverage and scope that we have, the more we can actually classify fish assemblages and understand what we would expect to find in most types of habitats. So a wetland or a beach, for example, on average, we can confidently say we should find the following different species. And if we visit a site and it doesn't have those, then we could look at some reasons or factors why that might be the case. So if there's been habitat modification or poor water quality, something like that, that could affect the reason why we don't see what we expect to see. So this data set is essentially allowing us to build up predictive abilities for predicting what we should find at certain types of habitats. So it's over about 66 species we've documented through this survey now, which is a lot of species. This map series just shows the expansion of this project over the years from 2015 to 2020. Again, the take-home message is that geographically it's expanded in scope from focusing mainly in 2015 around Cornwall and Akwesasne, and then now just expanding up and downstream from there. We've measured over 33,000 fish lengths. So again, this is another part of our data set. You can see this photo has a really nice image of a young of the year northern pike and a young of the year muskellunge side by side, and two species that we do find in wetlands of the St. Lawrence River. Overall, our project has documented a lot of, this is just showing 2020, but this is pretty characteristic of the 6-year time series. Most of the fish we find belong to the minnow family, and there's quite a diversity of minnows in the river, over 20 species. And we also find quite a few species of sunfish, and then several species of perch. So yellow perch is extremely common species that we find too. Almost all sites we go to have yellow perch. Unfortunately, you can see in that red pie segment there, round gobies are also a very common species. This is an invasive species that has shown up over the last couple decades in the St. Lawrence River. It's found at almost all sites we visit now, sometimes in very high abundance. Just to give you individual species, you don't need to look too deeply in that. Take-home message is if you're going to do a survey of the St. Lawrence River, at most sites you're going to come across a minnow species, the blunt-nosed minnow. It's extremely widespread, very common fish species of the small fish species of the St. Lawrence River. Of those 400 sites or 200 sites we've gone to, 70 of those have been wetland sites. So we have a really large, nice data set of wetland collection sites now. We've collected over 150,000, I should say we've documented 150,000 fish species at these 70 wetland sites. 150,000 fish from 60 species. And I've just shown some images here of some very common wetland fish you might come across. Yellow perch is a very common one. Small young of the year largemouth bass, the sunfish family, so pumpkin seed and bluegills. Some other here. So if you go to a wetland, this is a nice healthy wetland. We'd expect to find these species. I've also included round gobies. They are quite common in wetlands, unfortunately, too, the St. Lawrence River. They shouldn't be there, but they're, if you're going to sample a wetland, you're likely to find those as well. An exciting project component that we've added in 2020 is adding in the collection of water to filter and sample for environmental DNA. So this is where we have all species that inhabit aquatic ecosystems give off cells of their body. So they just kind of slough them off, or there could be genetic material that comes off them. And that's just floating in the water column at various concentrations. And there's a lot of factors that are involved with being able to actually find some of that DNA that's floating in the water. But in theory, you should be able to go scoop up a water sample, bring it back, and through some lab analyses, amplify certain types of genetic material and see what's actually in the water at your site. This is an exciting new tool that we're adding on in addition to our traditional sampling, where we physically go out and collect fish using nets, for example. So we're adding this on just to see if we can develop it as a tool first. We're in very preliminary stages of developing this tool, but the hope is that this will give us added information on tracking biodiversity. So for wetlands, for example, we could sometimes sampling wetlands can be quite difficult. There's the bottom substrates could be really soft, so the team could have trouble walking through that. We could also have a lot of plants, really thick vegetation that make it difficult to pull a net through a wetland. So if we're able to actually just collect a water sample, and if that can tell us what we need to know, then that's really valuable to us. It also reduces our handling of fish and our disturbance of the wetland or an aquatic site as well, if we can just grab a water sample. So it's a technique that we're developing. We're actually in the lab today doing this. So it involves collecting water in the field, and then you bring it back to your lab. You filter it through some really fine filters that trap all of the genetic material, the DNA in the water, and then you can extract that from your filter, amplify it through PCR. Very similar to what they're doing with COVID right now. COVID is just, when you do a COVID test, that's just a PCR test where they amplify genetic material from the virus to see if it's present or absent. So it's the same idea. We're trying to amplify genetic material for fish species. It could be individual fish species. It could be all fish species at a site to look at overall diversity from these water samples. And then ultimately, we want to use this as a presence-absence tool for looking at species at risk or invasive species. But we're working with some university researchers at Queen's University that have a lot of expertise in this field. So it's something we're developing at the Institute, and we're excited to move forward with this in 2021. You can see there's Amanda Nurse there. She's been leading the eDNA work at the River Institute. You can see a gel electrophoresis showing some results that we had for positive hits for different species there. Another area we're really excited about at the Institute is the use of drones. So you can attach different types of camera payloads to drones, and depending on the type of camera, you can get different information. So you can get a regular camera just in high resolution that gives you great imagery. You can take a series of photos and stitch those all together and make an overall map of your habitat. If you have other types of cameras like we do at the River Institute, so we have thermal cameras that give us information on temperature, and we also have multispectral cameras that collect information on different wavelengths of light, particularly infrared and near infrared, and those really help you view vegetation health really well. So chlorophyll pigments, for example. So I'll show you a few images of what that might look like. So the basic type of drone just gives you a nice overhead image of your site you're surveying. So in this case, this is the mouth of the Salmon River in Akwesasne, a beautiful wetland, huge, massive wetland of Lake St. Francis. And in this image, we can see terrestrial vegetation. We can see different types of vegetation patches there. We can also see aquatic vegetation patches from overhead too. This is all information that we would have to, before we had access to drones, we'd have to kind of measure this in the field by walking through it and using a GPS, for example, to waypoint things. So the use of drones just gives us so much more information and a really clear image of distribution. You can go back at this, look at this imagery, weeks, months, years later, and compare changes or do some more analysis on it. So this is a summer image of the mouth. We can, the nice thing about drones is that we can revisit sites on a seasonal basis. We can see changes in wetlands. You can see at the top there, there's a beaver lodge there, a nice collection of sticks and everything in the water in front of it that it's set aside for the winter. So again, nice to be able to compare seasonal changes. And if we use our multispectral camera, that gives us information on plant health. So the greener the image there, the healthier the plants are, the more chlorophyll is present. So you can see, you can kind of tell that a lot of those shoots that have been cut in front of that beaver lodge are new, but you can also see some logs in the river that are half submerged that are also really popping out in terms of the chlorophyll. So what that's really showing you is that there's algae attached to those logs, and it's doing pretty well. So even in the fall, some of these substrates where algae can attach to still have quite a bit of chlorophyll in them. Just another view here, so you can see an overhead view of our boat there. You can see some logs kind of half submerged in the water there. And if we switch to our multispectral camera, you can see those logs now have quite a bit of algae on those. So information you really couldn't see with regular imagery that now you can see with different wavelengths of light. So this is something we're trying to develop at the River Institute through image classification, and it's giving us a lot more information on aquatic vegetation at our sites. Despite COVID, our team was out in 2020, and we had a really successful field year, actually. We visited lots of wetlands in the river, and you can see the whole team wearing masks there. There's lots of adaptations where I'm anticipating we'll have to do in 2021 again as well. So things we want to keep looking at, we're expanding our use of drones. We flew, I want to say, about 10 to 12 sites in 2020 with our drones, with our new camera system. So we want to expand that and make sure that we can collect really high resolution information with different types of cameras, and then be able to do different image classifications on those to get more information about our habitats. So we could classify plant communities, for example. So here you can see Emily and Evan, two of our advanced drone pilots at the River Institute, just attaching some cameras to our larger drone. We're also getting more quantitative with our vegetation surveys. This is something that Dr. Marianne Perron at the River Institute is helping us with. You can see Marianne has a quadrat floating in the water there. So it's a standardized way of collecting information on what plant species are at a site. And then we can, in combination with our drone imagery, get really fine-tuned information on what plants are at each site. And then that helps us understand why we see different fish communities and species associated with different sites. The very first slide I showed you, gar, you're almost always going to find gar when you have wild celery and lily pads, for example, at a site. They love those two species of plants combined. So, but there's lots of different combinations of plant fish species matchups that you can really get into when you have really good quantitative information. We're using our drones and our survey also to get more information on the impacts of water level fluctuation. So this is upstream of the Moses Saunders Dam in Lake St. Lawrence by Hoople Bay. You can see later in the fall, water levels really draw down, and it has impacts on the quality of these types of habitats. So with drones, we're able to kind of get imagery on a periodic basis of these sites, and we can also visit them and perform our fish surveys and collect information on water quality, things like that, just to track how the impacts of water level fluctuations and how those are impacting fish species. Obviously, you're not going to find fish when a site is completely dried up like this, but 2 months prior to this photo, we were sampling at this site and finding quite a bit of fish. So allows us to get that perspective, seasonal change perspective. All the information we're collecting as part of this project is feeding into the Great River Report, which I believe you've seen a presentation on that earlier this week by Megan Mitchell. So it's a project, joint project between the River Institute and Mohawk Council of Akwesasne, being led by Dr. Lee Mugahi, and the information from FINS is feeding this in terms of being able to develop indicators for the health of the St. Lawrence River. So we've sampled different sections of the river, different types of habitats, and from that information we can generate indicators, good, bad, trends, up, down, things like that. So all this information will be available as part of the Great River Report as well, which is coming out in various formats fairly soon. Just to summarize the project, we've had lots of great impacts. We've, over this past 6 years, created one of the largest fish data sets for the Upper St. Lawrence River. It's a big thing. So this is allowing us to fill in critical knowledge gaps for species of risk and invasive species. It's a great training opportunity for people as well to come out in the field, learn how to use different instruments, even fly drones, for example, with some training. And we're developing these new technologies like eDNA and drone use as part of the project as well, which is just pushing the frontier of science in terms of this research. So yeah, I think I'm almost at 3 o'clock there. We're excited. As I mentioned, this is a 6-year project. We're going into our seventh year, and we're going to be continuing our eDNA research as well as our drone research. So I'm excited to keep working with the Mohawk Council of Akwesasne. And if anyone has any questions on the project, I just want to thank Emily Cormier for helping put together this beautiful presentation, all the different images and graphs, and all of our funders up to date as well. A lot of appreciation to them as well. So if anyone has any questions, happy to answer those.
I have a question, Matt.
Sure, is that Megan?
Yep. Hi. So out of all the fish you have captured and like identified, what would be one that you would consider as like rare almost, or that you don't really see a lot in the river here?
Good question. Grass peckerel is pretty rare. We don't catch a lot of those. Young of the year muskellunge are also very rare. I'm going based on how excited the team gets when we come across one of these, but yes, the young of the year muskie are very rare as well. I think one time we caught a lamprey that had been attached to a fish. So they're very, so the type of seine, the sampling equipment we use is a seine, and it just helps you target certain smaller fish that are relatively slow moving. And fish inhabit a whole range of water depths. So we could be missing some fish that are a little bit further offshore in deeper water, or at the bottom sometimes too. So of the 66 fish species we've caught, we know that there's American eels in the river, there's sturgeon, there's some other species that we're just not going to really come across with our sampling methods. We are adding some large fish sampling as well in 2021. So we'll be using some gill nets, for example. That'll help us maybe catch some more interesting fish. But anyway, the roundabout way of answering the question, I think really rare species would be things like grass peckerel and muskellunge.
All right, thanks for answering my question.
I'm pretty amazed. Oh, this is Amy from the St. Regis Mohawk tribe. At how many fish samples do you have collected? What constituents? What are you looking for in your sampling? Specifically mercury or a whole bunch of stuff?
We're not, so there's no contaminant analysis on we're mostly just documenting the distribution of these species. And once we catch them, we put the majority of them back in the river where we catch them. We have in 2020, we started kind of a tangential project with the MCA where we were starting to collect larger sport fish. And that's something that we're going to continue on in 2021, where we're collecting fish that people might consume, larger sport fish, and then taking tissues from those. But the main focus of the FINS project up to date has just been documenting the distribution of all these fish, their abundance, trying to find especially species at risk. There's just huge knowledge gaps on where they are in the river. And a lot of times what we're finding actually is that species that we thought are rare, if you just look for them and you know where to look for them, they're actually not as rare as we thought. So that's another valuable outcome of this. But I hope that answers your question. We are starting to get into some contaminant analysis, but that's mostly started in 2020, and that's going to be more focused on larger fish.
Yes, it does. Thank you.
Oh, Jeff has a question here. I know MCA is a partner on the project to develop the use of eDNA technology for communities. How do you see MCA and similar entities using the results? That's a good question, Jeff. So the eDNA is a very preliminary tool. You can imagine when you take a water sample and you're looking for the presence or absence of a specific species of fish, for example, a rare species or one that's at risk, and you go through the whole lab analysis and you get to the end and you don't find a positive hit on that specific species. Does that mean that the fish wasn't there? Possibly. It could also mean that just that one sample of water you took didn't have their DNA, but maybe their DNA was present a little bit further away or something like that. So there's all these different factors that impact the messaging you get from eDNA sampling. So that's why it's going to be something that we have to develop carefully and understand the limitations of it at the River Institute. So I don't think it's ever going to supplant our traditional sampling, but what I see people using the results, if we're able to do our traditional sampling and say we find a species of risk like pugnose shiner, for example, which is a species we focus on, if we can pair that with eDNA, where the eDNA results scooping up a water sample also give us a positive hit on that particular species we're looking at. And if we're able to repeat that at various different sites, then we've actually developed a really, really valuable technology and tool that can be used by other government agencies using the specific DNA sequence that we're focused on. So that could be of interest to provincial agencies, federal agencies that are also focused on species at risk. I think in terms of our working with Akwesasne, the eDNA allows us to get more information on the diversity of species in a habitat, because we can collect a water sample, and if we're able to sequence all the different genetic material in that water sample, we could actually, in theory, see how many fish species are present, invertebrates, plants, a whole suite of different taxa that maybe we might have overlooked with traditional sampling. So again, it's a pretty exciting tool, but we're very at the preliminary stage where we just need to understand how much we can, I don't want to say trust, but how much we can rely on the signal that we're getting out of that tool. If anyone has any other questions at all.
Hi, Matt. This is Matt as well from Thousand Islands National Park, and I'd say great presentation. I'm really amazed by the huge range you guys cover and the massive data set you've collected. Thanks, and Matt. The use of eDNA and drones at the forefront of that. I'm just wondering on the invasive species side, I know the report's coming out shortly, but are you seeing an increase of species in the last 5 years like goby and others, or how are those looking?
It's a good question. We've been doing this for 6 years, so I was a little hesitant to talk about trends over a relatively short time. Gobies, we are catching less of them at our sites on average, but it's not significant. It's something I've noticed over the last 6 years. We are seeing an increase, and we've actually published some research on this in the distribution of tube-nose goby, which are moving from Lake Ontario downstream towards Cornwall. And they're kind of at the halfway point now. They did about, I'm going to be presenting some of this research at the upcoming Angler Conference, but they did about an 80-kilometer increase in distribution last year from the previous year. So sites we were visiting in 2019 where we didn't see tube-nose goby, we saw them last year in 2020. So again, this project, it's a leading edge of it's really good timing for this project that we're actually out there because this is a brand new fish species moving down the river. We're also on the lookout for another invasive called tench, which is moving upstream from Montreal area. And we came across one tench in the Montreal area, but nothing in Lake St. Francis yet. But we're just kind of waiting to see. We know that they're in the area based on some adults that were caught and by some other organizations. So we're anticipating we'll probably start seeing those in our survey as well, which really shows the importance of, when you talk about invasive species, just collecting this baseline information of what this ecosystem looks like before they get in, so then we can really understand what changes are going to happen when they do get in. But plants as well, we do document quite a number of invasive plants in our survey. So Eurasian milfoil is probably the most common one we come across. And that has been increasing in some sites dramatically. In some cases, we couldn't even get our boat through some sites that we could previously just because of, fouling of the propellers. So, good question, Matt. Thanks.
So it looks like we don't have any other questions. I just want to thank you so much, Matt. That was a great presentation, and it was very aesthetically well done as well. I noticed that. It was very, very nice.
Thanks, Brittany. You can, I just wanted to give a shout out to Emily Cormier at the River Institute she helped put that together.
She did a wonderful job. It was very,
yes.
very engaging. Thank you. Okay, everybody. So it looks like we're going to take a 50-minute break, and we are going to have Clarkson University join us at 4 p.m. for the River Environment and Sensor Observation Network project. So we will see you all at 4.
All right, thanks.
Thanks, Matt.
Thanks, everyone. Thanks, Matt.
Thank you, Matt.